Right, these are the Unit 2 biology notes on DNA. Make sure you're filling out the document on Canvas as well. We're going to start by talking about DNA structure. So DNA stands for deoxyribonucleic acid. It is a type of nucleic acid. We'll talk about a second type as well. DNA is just one. The purpose of DNA is to store genetic information that's used to create proteins. So remember that DNA is one of the characteristics of life. So all living things, both prokaryotes, eukaryotes, plants, animals, bacteria, every type of cell is going to have DNA in it. DNA is made of 4 nucleotides that are going to pair in a very specific way. So we have adenine that's always going to pair with thymine. So we abbreviate those A and T. Those will always go together. And guanine is always going to pair with cytosine. So G and C always pair together. The way I remember this is that apples grow on trees and cars go in the garage. So A and T, C and G. So what is the structure of DNA? DNA has a very unique shape. It's called a double helix, which is similar to a twisted ladder. So if you think of these long strands on the side as sort of the sides of the ladder, then it has those bases in the center that make the steps of the ladder. And then it's like that ladder was twisted in sort of a spiral shape. So the sides of this ladder are made of phosphates and a type of sugar called deoxyribose. And the sides are just going to be alternating. So it's going to go phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way up on both sides. The steps of the ladder in the center are those nucleotide base pairs. So that's where we're going to have A and T bonded together or C and G bonded together. And the order of those bases, right, it might be T, T, C, G, G, whatever it may be, those steps are what's going to give DNA its unique information. So we also have another kind of nucleic acid called RNA. RNA stands for ribonucleic acid. It's another type of nucleic acid, and it's used in the production of proteins using those DNA instructions. It's slightly different from DNA. There's some unique differences, but it has a very similar structure. So RNA is going to be single-stranded, while DNA is double-stranded. So you can see in this picture here, RNA only has one of those backbones, whereas DNA has 2 of those backbones. So RNA is a single strand, DNA is a double strand. They're going to use different kinds of sugar, so you can see that right in the name. A ribonucleic acid, RNA is going to use ribose sugar. Deoxyribonucleic acid, DNA is going to use deoxyribose sugar. So remember, those are in the backbone, so it alternates phosphate, sugar, phosphate, sugar, phosphate, sugar, but the type of sugar may be slightly different. RNA has 4 nucleotides again that are going to pair in a specific way, but one of those nucleotides is different. So we still have guanine and cytosine that pair together, but instead of A and T, we have A and U. So adenine is going to pair with uracil in RNA. So notice A, G, C are both the same. DNA has thymine, while RNA has uracil. So here are the overall differences between DNA and RNA. DNA is double-stranded. RNA is single-stranded. DNA has adenine, guanine, cytosine, and thymine. RNA has adenine, guanine, cytosine, and uracil. DNA is going to be located in the nucleus of eukaryotes. RNA is going to be in the nucleus and in the cytoplasm. It's also going to be used to make ribosomes. In DNA, we use deoxyribose sugar in the backbone. RNA, we use ribose sugar in the backbone. All right, next section is on DNA replication and mitosis. So what is DNA replication? DNA replication is going to make an exact copy of all the DNA in the cell. So just like if we make a copy of a piece of paper, we're going to get another identical copy of it. It's exactly what we're doing in DNA replication. We have one set of DNA in the cell. During DNA replication, we're going to copy all of it to make a second set. This is going to happen before mitosis during what we call the S phase of the cell cycle. So here's the cell cycle. We have G1, S, G2, and M. So G1, the cell is going to grow. S, we think kind of the word synthesis. That's when the DNA is going to replicate. Then G2, the cell is going to grow again. And then M is where it's going to do mitosis, and it's going to actually divide and make more cells. So it's going to cycle through this, right? Grow, replicate, grow, divide. Grow, replicate, grow, divide, over and over. So here's more about the cell cycle. The cell cycle describes the entire life of a cell. So every cell is going to be in one of these different parts its entire life, right? It's going to cycle through these different stages throughout its life. There's 5 unique parts. So first we have G1. G stands for growth. So this is a growth phase when the cell is just going to be getting bigger. It's doing its normal cell function and it's growing. S, like we said, is the phase where DNA is going to replicate. Then we have G2, which is another growth phase. So it's growing again, but it has 2 copies of DNA. G1, it has 1 copy of DNA. G2, it's growing, but with 2 copies of DNA. Then we have M, which is when mitosis occurs, when that cell is going to replicate. And then if it's not in one of those 4, it's in something called G0, which is when the cell is just resting, right? It's doing its normal cell stuff, but it's not growing. It's not getting any bigger. It's not duplicating. It's not replicating, right? It's just doing its normal cell processes. So every cell is at an area of the cell cycle its entire life. So the 4 phases, G0, G1, S, and G2, are all part of something called interphase. That's where the cell is going to spend most of its life. So most of a cell's life is spent in interphase. Phase M is very short, and that's specifically when the cell is dividing in order to make new cells. So what is mitosis? We've mentioned that word a couple times, but what is it? The purpose of mitosis is to create 2 identical copies of a cell. So we're trying to just make a copy of the entire cell. So not just the DNA, but the whole cell. And this is going to occur for 3 reasons. To replace damaged or dead cells, right? If a cell dies or is damaged, then we got to replace that cell. To repair any injuries. So if you get a cut on your finger, your skin cells are going to replicate in order to repair that cut. And then 3 is to make the organism grow. As an organism gets bigger, it has more cells. So we need mitosis to replace any dead cells, otherwise we would just wither away and all our cells would disappear, right? To repair any injuries and to help the organism grow, to get more cells to grow. After that short M phase, the cell is going to go back into G0, where that cell is resting. So how is DNA packaged? Before mitosis can even happen, the DNA has to be condensed. This prevents any mistakes happening during division. During division, we need the DNA to be evenly split between 2 cells, right? We can't have more DNA in one and less in another. We have to evenly split it. In order to evenly split it, we got to make sure that this long string of DNA is packaged so that we can carefully move it to where it needs to go. So the first thing that's going to happen is that DNA is going to wrap around proteins, right? That long string of DNA is going to wrap around proteins and twist into coils called chromatin. So instead of being these thin little strands, now it's like a thicker, stronger strand. Then that chromatin is going to twist again to form chromosomes. As it twists, it's going to get shorter and more tightly packaged into what we call a chromosome. So humans have 46 chromosomes. Different organisms have different number of chromosomes. Fruit flies, for example, have 4 chromosomes. Humans have 46 chromosomes, right? Chromosomes are that condensed DNA that's all nice and packaged, easy to move around the cell. All right, so let's talk about what does DNA actually do. And what DNA does is something called protein synthesis. So protein synthesis is the process of using those DNA instructions to first make mRNA and then make a protein. So there's 2 steps in protein synthesis. The first step is transcription. The second step is translation. Both of those steps together are going to help us get from the DNA instructions to a protein. So let's talk about transcription first. Transcription is going to happen in the nucleus. The DNA is always going to be located in the nucleus for a eukaryote. That's where it's most protected. So because this involves DNA directly, transcription is going to happen in the nucleus, right by where the DNA normally is. It's going to begin with one segment of DNA. So not the whole DNA strand, just a section of it. And it's going to create an mRNA version of that information. So remember, mRNA stands for messenger RNA. So we start with this double strand of DNA, and we're going to make a copy, an RNA copy, specifically mRNA copy of one side of that DNA. So it's just like reading the instructions here and copying it down onto a piece of paper, right? These instructions are never going to leave the nucleus. The DNA is never going to leave the nucleus, but we're going to copy down the information we need, and this information can leave the nucleus and be used for other things. So the first thing that happens in transcription is the DNA is going to untwist. It's in that twisted ladder, remember? So it's going to untwist so that we can read those nucleotides in the center. The mRNA is going to read that strand and attach the complementary nucleotide. So remember, RNA uses uracil, not thymine. So if you look down here, here's our DNA with our nucleotides in it, right? You can read A, T, G, A, T, C, right? It's going to split apart. And then the RNA is going to copy one side of the DNA. And it's going to put down its complement. So opposite of T is going to be A. Opposite of A, if it was on DNA, would be T. But we're not copying it onto DNA. We're copying it onto RNA. So it's going to be U. So TAC, TAG becomes AUG, AUC. Right? So it's just the opposite of whatever the DNA says. That's going to make our RNA. That mRNA is then going to leave the nucleus and start the next step, which is translation. Okay? So let's look at this example. Okay? So if DNA has A, our RNA is going to have a U. If DNA has T, RNA has A. DNA has C, RNA has G. DNA has G, RNA has C. So if our DNA says TAC, the mRNA that goes with it is going to read AUG. CGA is going to become GCU. GGC is going to become CCG. So notice we're writing down kind of the opposite, the matching letter from what's on DNA. Just remembering to swap out the T for a U. All right, so here's transcription. Transcription is going to go from DNA to RNA. Once we have that RNA copy, then we can go on to the next step, which is translation. So what is translation? Translation is going to happen either in the cytoplasm of the cell or in an organelle called the endoplasmic reticulum. Translation is going to start with that mRNA. That mRNA that we made in transcription is going to be the beginning of translation. And we're going to use a ribosome in order to use the RNA to create a protein. That protein is going to be made of amino acids. So the first step of translation is that the mRNA is going to connect with a ribosome. The ribosome is like a little translation machine. Right? So the mRNA is bringing the instructions from the nucleus. It's connecting with that translation machine. Then the mRNA code is read 3 bases at a time. So those 3 bases are called a codon. Each codon is going to represent a different amino acid. So when the ribosome reads that codon, reads those 3 bases, it's going to figure out, oh, this is the amino acid that matches that, and the correct amino acid is going to be brought to the ribosome, right? Then it's going to read the next 3 bases and bring the next amino acid, and the next 3 bases, and the next amino acid. So eventually we're going to have this chain of amino acids that makes a protein. This is going to continue until there's a specific codon that means stop. And that's where the end of the protein will be, right? So we're starting with this RNA, ending up with this protein that's made of a bunch of amino acids connected together. So we're going to use this codon wheel in order to figure out what the amino acids are for our protein. So the way that we use this codon wheel, we're going to read our mRNA in sets of 3, right? So we're always going to divide it into 3. read them 3 bases at a time on the mRNA. For this wheel, we're going to start on the inside, the big pieces of the pie, and then we're going to work out. For example, G is going to divide it into this sector. Then A, I'll follow it to this sector. And then C would give me aspartic acid, right? GAC. And that would be the amino acid that that piece of mRNA codes for. You don't have to memorize this wheel. You will always be given this wheel so you can figure out your amino acids. You just have to know how to use the chart. So here are some examples, right? Do these along with me so you can understand how it works. Make sure you get the same answer. So here we have U, G, U should be cysteine, right? Starting in the center, working your way out. A, G, C is going to be serine, right here. And then U, A, G, notice it says stop. That means that's going to be the end of the protein. Translation is going to stop at that point, right? There's not an amino acid name stopped. It literally means stop. You're at the end, right? All right, so that's the end of your notes for DNA. We're going to do lots of practice on transcription and translation in class. Please make sure you submit your notes on Canvas when you are finished.